All right, let's dive right in. Today, we're going deep into our own DNA to tackle a really fascinating biological puzzle. We're going to uncover a hidden layer of complexity in our genetic code, a layer that, thanks to an amazing new tool, scientists are just now beginning to understand.
So get this, it's a fact that might just blow your mind. You have about 20,000 genes. You know what else has roughly that same number? A tiny, simple roundworm. So how does that work? How do we build brains and hearts and complex immune systems? We're talking trillions of specialized cells with basically the same number of genetic parts as that simple worm. That is the huge mystery we're unpacking today.
So here's our game plan. First, we'll dig into what I'm calling the gene recipe mystery. Then, we'll see why the picture of what's going on has been so fuzzy for so long. After that, we'll introduce the hero of our story, a new tool called ScIsoX, see what it found, and finally, look at what this all means for the future of biology. Let's do it.
Okay, so that whole worm versus human puzzle, how do we even begin to solve it? Well, the answer starts when we realize that our genes aren't just simple on or off switches. Oh no, they are way, way more clever than that.
The real secret is a process with a bit of a fancy name, alternative splicing. But don't worry, the idea behind it is actually pretty simple. It's basically a biological copy and paste system that lets a single gene produce multiple, slightly different versions of a protein. And each of these different versions is called an isoform.
You know, the absolute best way to picture this is to think about a recipe. Imagine your gene is the master recipe for, say, a chocolate cake. Well, alternative splicing is what lets the chef create all sorts of variations from that one master recipe. You could make a gluten-free version, a low-sugar version, maybe an extra dark chocolate version. They're all still chocolate cake, right? But each isoform has a unique twist. And that is exactly how our cells take those 20,000 master recipes and create this incredible diversity.
So this is the big takeaway here. We have to completely update our mental model. The old textbook idea was pretty straightforward. One gene makes one protein. Simple. But the new reality is so much cooler. One gene can actually make many different proteins, or isoforms. It's not a simple light switch. It's more like a sophisticated control panel with tons of different knobs and sliders, giving our cells an amazing number of options.
Now, you might be thinking, if this has been going on inside of us all along, why is this just now becoming clear? Well, it turns out that actually trying to see all these different isoforms in action has been a massive, massive technical headache for scientists.
It really boiled down to three huge challenges. First, the data was basically a mess. Imagine trying to find a specific document in a giant, disorganized pile of papers. That's what it was like trying to figure out which isoforms belonged to which gene. Second, the files were memory hogs. The obvious way to store this information created these gigantic files that were mostly, well, empty space. It was incredibly inefficient. And third, the right tools just didn't exist. There was no good systematic way to measure and ask the most basic questions about all this complexity.
And that is where our hero, a new computational framework called ScIsoX, comes into the picture. It was built from the ground up to solve these exact problems and finally bring this hidden genetic world into sharp focus.
Think of ScIsoX as a complete toolkit that tackles all three of those headaches head-on. It's got a revolutionary new filing system that organizes everything perfectly. It gives scientists a brand new dashboard with 7 key metrics to finally put numbers on all this complexity. And it has powerful visualization tools so you can actually see the patterns for the first time.
Now, these 7 metrics are really the heart of the whole thing. They let scientists ask these incredibly precise questions, like how many different isoforms is a gene using inside just one single cell? Or how different is that from one cell to the next? And are some isoforms used only by specific types of cells, like a neuron versus a skin cell? These are the kinds of fundamental questions ScIsoX was built to answer.
So that's the tool. Pretty cool, right? But what happens when you point this powerful new lens at our own biology? Well, the secrets it started to uncover are just fascinating.
The researchers pointed ScIsoX at 3 totally different biological systems. 2 were developing systems, mouse blood and brain, where cells are constantly changing and figuring out what to become. And the third was a stable, specialized system, our own human immune cells, which already have very specific jobs to do. And right away, they saw this incredible pattern emerge.
In those developing systems, where cells are still specializing, the use of different isoforms was kind of all over the place, really flexible and varied. But in the specialized immune cells, the pattern was much tighter. It was focused and it was clearly driven by the specific job each cell had to do.
This really gets to the heart of it. This quote from the paper says it all perfectly. In these specialized immune cells, that variety we see isn't just random biological noise. No, it's a highly controlled system that's directly tied to a cell's function. In other words, this complexity has a purpose.
To make this super concrete, let's just zoom in on one single really important gene. It's called MS4A1, and it's absolutely critical for our B cells. Those are the immune cells that make antibodies. This gives us a fantastic snapshot of that complexity in action. Take a look at the B cell. See how it uses high amounts of isoform A and B? But a T cell, it uses a completely different cocktail. And a monocyte, another immune cell, uses yet another mix. So it's not about one isoform being simply on or off. It's the specific combination, the unique recipe of isoforms that defines what that cell is and what it can do.
Okay, so we've seen the puzzle, the tool that solved it, and the incredible discovery. But let's zoom all the way out. Why does this new level of detail matter so much for the future of biology and even medicine?
Before we look ahead, let's just quickly lock in the key takeaways. First, one gene is like a master recipe that can be sliced and diced into many different variations called isoforms. Second, this hidden complexity isn't random, it's tied directly to what a cell does. And finally, powerful new tools like ScIsoX are letting us see and measure this hidden biological world for the very first time.
And just to really drive home what a huge technical leap this is, remember that old clunky way of storing the data? It would have created a file that was over 98% empty, wasted space. ScIsoX's clever design completely sidesteps that problem, which is what makes this kind of analysis possible at all.
At the end of the day, what this all means is that we can now study life at a resolution we have never, ever had before. We're essentially adding a whole new, incredibly detailed dimension to our map of the human genome. And the future applications are just huge. We can better understand how a single cell develops into a complex person. We can start to pinpoint exactly how errors in this splicing process lead to diseases like cancer. And maybe most excitingly, we can start designing smarter medicines that target only the one bad isoform of a gene, while leaving all the healthy versions to do their jobs. And that really brings us to the end. Being able to see this hidden layer of our genetic code isn't just the answer to an old puzzle, it's really the beginning of thousands of new questions. It makes you wonder, right? What other amazing biological secrets are hiding in plain sight, just waiting for the right tool to bring them into the light? Thanks so much for tuning in.